Your question on enzymes can be slightly tricky because they will often give you graphs and then they will ask you to describe the graph and explain the graph and all of that. So that is something we have to practice answering. So the first question part is easy. Enzymes are catalysts. Describe what is meant by the term catalyst. So can you just give me the definition of catalyst? What are catalysts?
Catalysts are is a substance that helps like it makes the chemical
Yeah.
reaction faster.
Yes. So it's going to make the chemical reaction go faster, and uh, and it's going to make the chemical reaction go faster and without changing themselves. That is very important, right? So they will not change themselves, but they make the chemical reaction go faster. Okay, enzymes are proteins. State the name of chemical elements found in all proteins. So, like elements are, like iron, zinc, copper, these are elements. So, what are the elements we find in proteins?
Uh, Carbon,
Yes,
um,
correct. And
hy- hydrogen, and oxygen.
yes, that is correct. Hydrogen, oxygen, and also nitrogen. Okay.
Okay.
Proteins also have nitrogen in it. Okay. Lipase is an enzyme found in human digestive system. Figure 1.1 shows the effect of pH on the percentage activity of lipase. Describe and explain the changes in the percentage activity of lipase, and this is for 6 marks. Okay. And this is, I believe this question came this year in your 2024 paper. So, um, over here, this is the graph of pH against your activity of the enzyme. So you need to explain first of all this question. Hajra says both describe and explain. So describe means that first you describe the shape of the graph, that the shape of the graph is like this, that as the pH is increasing, the activity increases, all of that. Okay. And then you need to explain why. So you need to relate that to the topic of denaturing and the shape of the active site and all of that. So this is for 6 marks. So at least 2 points preferably on the description and then 4 points on the explanation as to why. So you can take your time, take 3-4 minutes and type out the answer for this. So the reason I'm going over this topic with you is because these questions come with a lot of marks. 6 marks, 5 marks, 4 marks. So you need to score as much as you can on these questions because they will really help you score a good, like really high marks.
As for the starting of the reaction, like when it goes up, uh, will it be that the enzyme has an optimum pH and then
Yes.
it will go to the highest rate of reaction?
Yes,
And
over here, optimum pH of around 7, right? So you want to mention
yeah,
that optimum pH of around 7. And if you want, you can link this to the topic of digestion as well. Lipase is found, for example, in the pancreatic enzymes. So you can relate it to the pH of the small intestine.
I know the answer for it, but I don't know how to phrase it properly.
Doesn't matter. Go ahead. Say your answer, and I'll help you with the phrasing. That's no problem.
Yeah, so the lipase, it will react with the pH, and then at 7 it will be at its highest rate of reaction.
Mm-hmm.
And then because the pH will be too high, it will the graph will show it coming down at till 10.
Yeah.
Uh,
Mm-hmm.
and then because it is too high, and the um enzyme will denature,
Yes.
and the shape of the active site will change, and it will not be complementary
Yes.
to the substrate.
Yes. Yes. Okay. Perfect. Good. Now I will just add. I'll take your answer pretty much, Hajra, and I'll just add a little bit to it. Okay.
Okay.
So the way you're going to answer this is this: as pH increases, the percentage activity also increases. The percentage activity also increases. Okay. So until it reaches optimum pH of 7, at which the activity of the enzyme is 100%. Okay. As the pH increases after the optimum pH, or as it increases further, the activity of the enzyme starts to decrease again. So this is the description of the graph, right? The first thing you should do is always, always, always start with just the description of the graph. Okay. So as the pH increases, the percentage activity is increasing until we get to the optimum pH at which the activity is 100%. After which the activity starts to decrease once again. So you first thing you always do in questions like this is start by just describing the graph. What can you see? Right? So this is the graph described. Now we will try to do explanation. Okay? Hi, Aljohara, how are you?
I'm good.
Okay, Aljohara, we're just doing this question right now. So our question was this, that lipase is an enzyme found in the human digestive system. Figure 1 shows the effect of pH on the percentage activity of the enzyme lipase. So we had to describe and explain both things, the change in the percentage activity of lipase. And this is for 6 marks. And I think this paper came this year in your 2024. They had given this question. Okay. So that's what I said, that we're going to be practicing these graph-like questions because they normally come with a lot of marks, 4 marks, 5 marks, 6 marks. So it's important that we answer these questions well because they will help you score really good marks or really high marks. So this is what we have got so far in the terms of our answer. This is how we're going to start our answer. First, just the description. Okay, so our description is that as the pH is increasing, the percentage activity is increasing until we get to the pH of 7. At the pH of 7, the activity is 100%. So this is the optimum pH. As the pH continues to increase after 7, the activity starts to decrease again. So this is the description. Now we have to give an explanation. What do we write down in the explanation? In the explanation, we have to answer the question like this. So in the explanation, Hajra, now your explanation, Hajra, by the way, was fine, but just how can you make it one step better? So over here, what you're going to say is that when the pH is low or the pH is high. Okay, the structure of the protein or the bonds in the protein, you can say it like that, are disrupted. Okay? This changes the shape of the active site and hence the this changes the shape of the active site and hence the enzyme or the enzyme can no longer bind to the substrate. Okay, it can no longer bind to the substrate. Hence, the percentage activity will decrease as the enzyme is denatured. Okay, so the first point is your description. So divide this like this, that almost half the marks, 3 marks are for description and 3 marks are for the explanation. So your description is going to be that as the pH increases, the percentage activity increases until optimum pH at which maximum activity is seen. And as the pH starts to increase, the activity will also decrease. So explanation, description, then the explanation. Now this is true whether the pH is low or whether the pH is high. Okay, in both cases, if you're at low pH or high pH, the bonds of the protein are disrupted. Since the bond are disrupted, so this changes the shape of the active site. So the enzyme can no longer bind to the substrate well. Okay, so the percentage activity decreases because the enzymes are denatured. So it is important that you include these points. Is this making sense? Al-Johura, Hajra, all clear now?
Yes.
Okay.
Yes.
Hajra, it's important, Hajra. So this is how you're going to format it. So what you had said was perfectly fine, but this is how you'll put all the information together to make it like a proper 6 mark answer. Okay, let's take a look at another question. Give me 1 second, guys. Okay, so our question says a student investigated the effect of lactase Lactase is an enzyme. Okay. Wherever you guys see ASE, ASE is for enzymes. So effect of lactase on three different liquids: milk, lactose-free milk, and sucrose solution. So lactase is an enzyme that breaks down lactose. Okay. And people who are lactose intolerant, right? Like you people who can't drink milk and stuff, they don't have this enzyme in their body, lactase. So the student used an indicator to test for the presence of glucose. A sample of each liquid was tested before and after the treatment with lactase. So, for example, in milk, these are the results. Okay. The indicator turned brown in the presence of glucose, right? If there is no glucose, then the solution will stay blue. So in milk, the lactase changed color. The solution changed color. It went from blue to brown, meaning that glucose was produced. In lactose-free milk, the solution was brown at the start, meaning that at the start it also had glucose and towards the end it also had glucose. In sucrose solution, there was blue at the start and blue at the end, meaning that there was no glucose whatsoever. Okay. Now, explain the results of for the three liquids shown in table 4.1. And this is for 3 months. So in milk, you can see that color is changing place, meaning that before we did not have glucose and then we suddenly got glucose. In lactose-free milk, there was glucose at the start and glucose towards the end. In sucrose, there was no glucose at the start or at the end. You have to explain the results, and this is for 3 marks. Take a minute, think about the question in front of you, take 1 to 2 minutes, and think about it. How should we answer this question? Can you give an explanation as to why we see the color changes that we see? Even if you can't figure out all of them, but you can just figure out one or two, that is fine, right? Whatever you guys can figure out, think about it and tell me, why do you think we're seeing the results that we are seeing? All right, what do you think? Why am I, Hajra, what do you think? Why is it that in milk, the color is changing from blue to brown? So initially, the color is blue, meaning that there is no glucose. And then the color becomes brown because there is glucose. So think about what the enzyme is doing.
For the first one, for the milk, will it be that lactase will break down the glucose?
Sorry, Hajra, did you say something? I couldn't hear you. Okay, I will explain it again, Al-Johra. Yeah, Hajra, what did you say?
I said for the milk, will it be that
Yeah.
lactase breaks down
Yes.
the lactose in milk,
Yes.
like
Yes.
glucose, right? Breaks down
Yes.
the
Exactly.
something like that.
Exactly. Yes, yes, you're right. You're right. You're absolutely right. So basically, what the question is saying is that we have three solutions. Okay. One is just pure milk. Okay. Then we have a lactose free milk. And then we have a sucrose solution. Al Johora, sucrose is a sugar. Okay. It's different than glucose, but it's just a sugar. So the thing over here is that we have an enzyme. This enzyme is lactase. Okay. The enzyme lactase will break down the what does milk have? Milk has lactose inside it. So this enzyme lactase, it will break down the lactose to give us glucose. Now, when we have the milk, right? When we have milk, in milk there is lactose present. Since the lactose is present, so what will happen that when I will add the indicator, Al Johora, the indicator tells you the presence and absence of glucose. If glucose is present, you will get brown color. If the glucose is not there, you will get blue color. So I have got three solutions. In every solution, I am adding the enzyme and then I'm adding the indicator. So first I take my solution, let's say milk. I add the indicator and I note down the color. Then I add the enzyme and then I note down the color again. Okay, this is the process. Now, in milk, we have lactose. Since milk has lactose inside it, so what will happen? That at the start, the color will be blue. Why? Because before I add the enzyme, there is no glucose. Before I add the enzyme, there is no glucose. If there is no glucose, the color will be blue. Okay? Then I add my enzyme, lactase. This enzyme will break down the lactose into glucose. So if glucose is there, the color will become brown. Okay, so in solution number one, what is happening? That lactose is found in solution number one, lactose is found in milk. Okay, this enzyme, so when I will add my indicator, it will give me a blue color because there is no glucose, there is lactose. Then you add the enzyme. When you add the enzyme will break down the lactose into glucose. So the color will change. It will become brown because now glucose is present. Then in lactose-free milk, see, the color is brown because it does not have any lactose. This does not have any lactose. What does it have? It has glucose. Okay, so lactose-free milk is made up of glucose, right? That is what we can see. This is something that you will not obviously know yourself. This is something that you will have to figure out looking at the question. So in the question, you can see that we have brown color. Before it's also brown and after it is also brown. So what does that tell us? That in lactose-free milk, it must be glucose. That's why before I add the enzyme and after the add the enzyme, the color is the same. It is brown because there must be glucose present. Okay. Then in sucrose, the color remains blue. Blue is the absence of sugar, okay? Or the absence of glucose. So sucrose has no glucose inside it, okay? And since this enzyme is for lactose, this enzyme is for lactose. Since the enzyme is for lactose, it will not work on sucrose, right? Because what did I tell you guys about the enzyme? Enzymes are very specific. So if lactase works on lactose, it won't work on sucrose. It won't break down sucrose. So sucrose will give you the same blue color. There is no sugar, there is no glucose, right? If I want to break down sucrose, I have to add the enzyme for sucrose. I can't use the enzyme of lactose, right? So sucrose will give me no color change because there is nothing happening. Okay, there is nothing happening in sucrose. Why? Because the enzyme is specific for lactose. This is the enzyme for lactose. It's not going to bind to sucrose, right? So no color change in sucrose. In lactose-free milk, again, the color is brown. Why? Because the in lactose-free milk, there is no lactose, right? But in normal milk, we have lactose. So when I add the indicator, it will initially give me blue. Why? Because there is no glucose. But after I add the enzyme, the lactose will be broken down into glucose and the color change will be visible. Right? Making sense now? Johara, it's making sense now what the question is asking you?
Yes.
Ajra, making sense now?
Yes.
Aljora, is this making sense what the question is asking you? Do you want me to go over it again?
No, it's fine.
Okay. Are you sure? All clear? Okay. The student kept the solutions at a temperature that was close to the optimum during the investigation. Using your knowledge of the effect of temperature on enzyme activity, explain why this was important. So, while the student was doing the experiment, he kept everything at optimum temperature. So, you guys have to explain what is the question asking you to basically explain the effect of temperature on enzyme activity. That's what they're asking. Do you guys remember in the notes in the class, I told you a whole paragraph of what is the effect of temperature on enzyme activity? What will happen if the temperature is too low? What will happen if the temperature is too high? So take the notes. You guys can write down the answer I gave you in the class. I don't mind. But you guys have to explain the effect of temperature on the activity of enzyme. What will happen if the temperature is too low? What will happen if the temperature is too high? And this is for 4 marks. Write down whatever you can remember. Even if you guys can't remember the entire thing, I'm okay with it. Whatever you guys can remember, that is okay with me. All right. So what is the effect of temperature on enzyme activity? All right. So is anybody done yet, Hajra? Al-Juhura, what do you guys think? How should we answer this? What is the effect of temperature on the enzyme activity?
Also,
Yeah.
when the rate of reaction reaches the maximum value, that is the optimum pH.
Yes. Optimum
And
temperature.
then,
Yes. This is about temperature.
yeah,
Yes.
optimum temperature. And if the temperature it increases or decreases, the rate of reaction will also start to fall.
Yeah.
And
Yeah.
then the active site will denature.
Yeah. Yes.
And
Correct.
then, lactase will no longer be complementary to lactose.
Yes. That is correct. Good. Fine. Al-Juhura, what do you think? How should we answer this? Al-Juhara, any idea? Is there anything that you would like to add to the answer? Okay, so okay, the answer that we're going to give over here is this: as temperature increases, the rate of reaction or the rate of or the enzyme activity increases. First, this is the first we have to give an explanation. Okay, as the temperature increases, the enzyme activity increases. Why? The reason for this is because as temperature increases, the kinetic energy of the enzymes increase. So there will be more collisions, and since there will be more collisions, the rate of enzyme activity will be high. Okay, this is why when we are talking about our increase, so when you're giving the answer, you have to explain it in two ways. First, talk about what happens when the temperature increases. So as the temperature increases, the enzyme activity also increases. Let me correct this. Also increases. This is because the kinetic energy of the enzymes increase. So there will be more collisions because the enzymes will move faster. So there will be more collisions. So the rate of enzyme activity will be high. As the temperature reaches maximum temperature, or I'm saying maximum temperature, I meant optimum temperature, the enzyme activity will be maximum. As the temperature goes further, or as the temperature increases further, as it increases past the optimum temperature, the rate of activity or the enzyme activity decreases due to disruption of the bonds of the protein. This changes the shape of the active site. So enzyme can no longer bind to substrate efficiently, and the enzyme is denatured. Okay, this is going to be your answer. You have to explain both the points. You guys have to explain what happens as the temperature increases, and also you have to explain what happens as the temperature decreases. Sorry, as the temperature increases towards the optimum temperature, and then what happens when the temperature increases after the optimum temperature. For both the points, there is a different logic. In pH Hajra, you can talk about denaturing before the optimum pH and after the optimum pH. But in temperature, the logic is different. Before the optimum pH or before the optimum temperature, the problem is not the denaturing. Before the optimum temperature, the problem is that the kinetic energy of the enzymes is less. So they will not be colliding. So there will be less product or there will be less activity. So the reason of why we see the change or the reason for change in activity before the optimum temperature and after the optimum temperature is different. For pH, you can say denaturization before and after the optimum pH. But in temperature, before and after, your answer will be different. Okay. So before the temperature in before the optimum pH, as the temperature will increase, the kinetic energy will increase. So more collisions between the enzymes and the substrate. Okay. So rate of activity will be high. Then the temperature will reach optimum temperature at which we will see maximum activity. As the temperature increases further, the enzyme activity will decrease. Why will it decrease? Because now the temperature is too high. It will disrupt the bonds of the protein. Okay. This will disrupt the bonds of the protein, and as a result it will change the shape of the active site, so the enzyme can no longer bind to the substrate efficiently, or it can no longer bind that well. So now the enzyme is going to be denatured. Okay, so Hajar, your answer was perfectly fine, but this is my recommendation that when giving the answer for before the optimum pH, you need to talk about kinetic energy and collisions, and then after the optimum temperature you can talk about the denaturization. Okay, in pH, it's the same, but in temperature, it's going to be slightly different. Right. Al Johara, your answer is good. Your answer is perfectly fine. Okay. So good. I am glad you guys have got the answer down. Good job, both of you. Okay. Let me share another question with you guys. Just give me 1 second. Okay, so we have this question that Hajra, you and I did the first part of this question previously. Okay, so now the second part of the question that I want you guys to do is that the action of lipase is affected by the temperature. On figure 1.2, show the axis for the graph for the effect of temperature on the activity of lipase. You have to complete the graph. Okay, so if you guys remember in the class, I made for you guys the graph for enzyme, temperature, as well as the pH. So you have to complete the graph by drawing a line to show the expected effect of temperature on the activity of lipase. The very complicated wording simply means that show the graph. That's it, just like I've taught you guys. Add a label line and a label to show the point at which all the lipase has been denatured. So at whichever point you feel like all the lipase has been denatured, just mark that point and label it that lipase denatured. Okay. So you have to draw the graph exactly like I drew for you in the class. So if you guys prefer do annotating on the screen, you can, or my advice to you is take out a piece of paper and your pen, pencil, whatever you like. Make a graph that shows what is the effect of temperature, and then label the point where the lipase has all been denatured. Okay, and you guys can send me the picture on WhatsApp or the Zoom chat, whatever you prefer.
So we have to draw the same as
The graph.
you showed us in
Yes,
class.
exactly.
Okay.
All right, Al-Johara, I got your graph. Your graph is perfectly fine. Your graph is great. Can you just mark on the graph the point where all the lipase will be denatured, right? So, like you in your previous answer, you explained that when the temperature is high, the enzyme will be denatured, all of that. So, just on the graph, make a dot when you feel like okay, now the enzyme is all going to be denatured. Yes, that's perfect. Good job, Al Johara. That is fine. Okay, Haja, I am just waiting on yours. Okay, now Al Johara, meanwhile, while Haja is drawing that, take a look at this. Explain why lipase cannot be used to break down or cannot be used to catalyze the breakdown of proteins. The enzyme lipase is for lipids. Okay, the enzyme lipase, Al Johara, is used for lipids. So they are basically saying, why can we not use it to break down proteins? So once again, you need to talk about, your active site, shape of the active site, all of that. Okay, you can use the answer that I gave in the class as well. You can use my own wordings. I don't mind. Okay, Hajra, you did not make the point of where the enzymes will be denatured. At which point of the graph will the enzymes be denatured? You have to add that point. Otherwise, your graph is okay. And also, Hajra, do take a look at this question as well. Why can lipase not be used to break down proteins? Did anybody figure out the answer for this one? Al Johara, did you get the answer yet? Hajra? Why can't I use the enzyme lipase? Why can't I use it to break down proteins?
Uh, because the only substrate for life is fat.
Yeah, why? Now you have to explain why. So you have to explain regarding, the active side, all of that, the lock-in key model, that you have to link it to that concept. Okay, Al Johara is saying she sent the answer on WhatsApp. Hmm. This enzymes using lipids and proteins is not complementary to protein. This enzymes. Yes. Aljora, your answer is fine. I'll just add a bit more to it in a minute. But yes, your answer is mostly correct. Good job. So, Hajar, you need to relate it to the concept of your lock and key model, and you need to link to the concept of the active site.
Oh,
So,
so
yeah, go on.
can I say that the shape of the active site is not complementary to protein,
Yes, correct.
and the
Yes.
shape of fat will be complementary to it?
Yes. So you are going to say over here that the enzyme lipase is specific for lipids only. The shape of the active site is not complementary to the shape of the proteins. Hence, proteins cannot bind to the lipase enzyme. Your hence, your proteins cannot bind to the lipase enzyme, so your enzyme substrate complex will not form, and hence the reaction will not happen, or the proteins will not be broken down. Okay, so this is related to that, to the entire lock and key theory. So lipase are specific to lipids. If you add the concept of specific, Al Johra mentioned that in her answer, so that is good. Okay, specific is it's like a proper term. So they're specific to lipids, and the shape of the active site is complementary to lipids, or it's not complementary to proteins, so proteins cannot bind with the enzyme to form the enzyme substrate complex. Okay, that is going to be your answer. All right, good job. I am happy. Al Johara, Hajra, both of you gave good answers today, so I am happy. Okay, so we're going to go ahead and we're going to stop it over here for today. Now, in our next class, we're going to go ahead and start off with our topic of circulation in humans. Okay.